Hello students, welcome to today's lesson on cause and effect in scientific experiment. In this lesson, we are going to take a look at variables like independent variable, dependent variable, controlled variable, and confounding variable. What do we mean by cause and effect? Scientific experiments always try to establish cause and effect since science rely on scientific experiment to discover new knowledge and skill. Science is the way of finding out if one event actually causes another event. This means that scientists try to prove that a change in one factor brings about a change in another factor. The factor that scientists change or manipulate is independent variable. The factor that scientists measures to see if it changes when the independent variable is changed is called dependent variable or effect. The first independent variable is the cause. Scientists always want to find out if changes in the independent variable or cause brings about a change in the dependent variable or effect. First, we have to describe variable. Variable is any environmental factor or stimulus or events that can affect all living organisms in a given ecosystem. To see the interaction of variables in a given ecosystem, let us take an example here, tomato seed germination. In tomato seed germination, the independent variable is the presence or absence of tomato juice that might affect germination of tomato seeds. You see, presence or absence of tomato juice here is the cause or independent variable. The dependent variable in this experiment is number of tomato seeds that gets germinated. The third variable is controlled variable. In our example, tomato seed germination, the controlled variable must be temperature, light intensity, number of seeds per dish, and volume of liquids added on the seed. I think we need to describe what's controlled variable. Controlled variable is simply the factor other than the independent variable that must be kept constant or controlled to avoid its influence on results. When these environmental factors or stimulus gets controlled, the only cause for the existence of experimental results must be the independent variable. In our example, the presence or absence of tomato juice. To prove that it is a change in the independent variable that are causing changes in the dependent variable or result or effect, we must take all the steps or to make sure that the experiment is a fair test. After we confirm that the experiment is fair, we must make sure that anything other than the independent variable or the cause which could affect the results kept constant or controlled. When other factors other than the independent variable gets controlled or constant, the only results or effects or dependent variable gets happen in our experiment is due to the independent variable, changes on the independent variable. In tomato seed experiment, as we mentioned before, controlled variables include temperature, light intensity or lighting conditions, number of seeds per dish, the volume of liquid added. All these environmental factors or variables must be constant. When these variables kept constant, the only change on the dependent variable or results happen due to change on the independent variable or cause. Occasionally there is a variable that might influence the results that we cannot control. These variables, what we call confounding variables. This confounding variable gets confound or confuse us to interpret the results in real or correct ways. In this case, we couldn't be certain that it was the independent variable or confounding variable that produce results or changes in the dependent variable. For example, if we want to measure carbon dioxide uptake by wet plants, as the light intensity changes over the day, we cannot control the effect of change in temperature. In this case, rate of carbon dioxide uptake might be influenced by the increasement of temperature and the availability of carbon dioxide concentration. In this viewpoint, we get confused that what affects the efficiency of carbon dioxide uptake is that availability of carbon dioxide or the increasement of temperature. So here, temperature that comes due to the increasement of light intensity could be a confounding variable. It confounds the effects or results of carbon dioxide uptake.